Welcome to today's deep dive. So, usually when we talk about modern medicine, you kind of expect a precision strike.
Right.
Like you have a bacterial infection, you take a pill, and that pill acts like a heat-seeking missile, right?
Ideally, yeah.
Yeah, just destroying the invaders while leaving your own body completely untouched. But what happens when that missile is so powerful that it accidentally starts taking out the load-bearing walls of your own house?
It's a terrifying thought.
It really is. And we are unpacking a massive stack of pharmacological research for you today to explore that exact scenario. Because our mission today is to understand a class of drugs that perfectly captures this uh, powerful but risky paradox of modern medicine. And those are fluoroquinolones.
Yeah, and to really understand fluoroquinolones, we have to look at what they are structurally. They are broad-spectrum, fully synthetic, antibacterial drugs. And, I mean, that distinction matters right away.
Fully synthetic, meaning they aren't, like, found in nature.
Exactly. If you look at penicillin or, a lot of the classic antibiotics, they were essentially harvested biology. We found them in fungi or soil bacteria.
Right. We sort of borrowed them from nature.
Yeah, exactly. But fluoroquinolones, on the other hand, are engineered chemistry. They're built from scratch in a laboratory setting to do a very, very specific job.
Wow. Okay.
And because they're so effective, the way we define them has undergone this massive clinical shift over the decades. I mean, historically, a doctor would define them by how they work as DNA gyrase inhibitors.
Right, focusing on the mechanism.
Right. But today, many physicians and regulatory bodies like the FDA, they often define them primarily by their severe toxicity profile.
Which is wild.
It is. They are basically known as the antibiotic class, with disabling side effects first and bacterial killers second.
It is such a rare and honestly wild trajectory for medication to take. So we're going to explore how an accidental discovery became a blockbuster drug, why doctors started prescribing it for basically everything, and how it eventually turned into this massive regulatory cautionary tale.
It's quite a story.
It is. And to understand how they became so ubiquitous, I was looking at the timeline in our research, and it seems like the entire origin of this drug class was a total accident. We're talking about, what, 1962, right?
Yeah, 1962, there was a chemist, George Lesher, working at the Sterling Winthrop Research Institute. And he wasn't looking for a new antibiotic at all.
Wait, he wasn't?
No, not at all. He was actually working on purifying chloroquine, which, is a really well-known anti-malarial drug.
Oh, right.
And during that chemical synthesis process, his team generated a contaminant. I mean, it was literally a waste byproduct of the reaction they were trying to run.
See, I feel like most people would just, throw that flask in the sink.
Oh, absolutely.
But his team actually examined this waste compound and realized it had some modest antibacterial activity. Specifically, the notes mention it worked against gram-negative bacteria. And actually, before we go further, what exactly does gram-negative mean for, someone listening who doesn't spend their weekends looking through a microscope?
Yeah, that's a great question. So bacteria generally fall into two main categories based on their cell wall structure. You have gram-positive and gram-negative.
Okay.
Gram-negative bacteria have an incredibly tough, lipid-rich outer membrane. It acts like a suit of biological armor, really.
So they're the hard ones to kill.
Exactly. It makes it notoriously difficult for a lot of drugs to physically penetrate the cell to kill it. So finding a compound that could just punch through that armor was highly intriguing for them.
I bet.
Yeah. So Lesher's team refined this waste byproduct into a drug called nalidixic acid. And they patented and marketed that in 1962.
But looking at the clinical data from the 60s and 70s, I mean, nalidixic acid wasn't exactly a miracle drug, was it?
No, not quite.
It seems like it was pretty weak.
Yeah.
The sources show it had terrible tissue penetration, it was metabolized incredibly fast, and really only reached effective concentrations in the urine.
Yeah, that was its main limitation.
So for almost 2 decades, doctors used it almost exclusively for uncomplicated urinary tract infections. It feels like it was just this niche, minor tool.
It was. But that changed dramatically with the medicinal chemistry revolution of the early 1980s.
Yeah, the 80s.
Right. Researchers in Japan and Germany, specifically at Kurin Pharmaceutical and Bayer, started systematically tinkering with the molecular structure of that original compound.
Just moving atoms around to see what happens?
Pretty much. And they made 2 critical modifications. First, they added a fluorine atom at the 6th position of the core ring structure. And second, they attached something called a piperazine ring.
Okay.
And this birthed the true fluoroquinolones, leading to drugs like ciprofloxacin or Cipro, in 1987.
Okay, let's unpack this, because this is where a minor UTI drug turns into a systemic powerhouse. A piperazine ring, I mean, that sounds like a component from a sci-fi spaceship.
It does sound pretty futuristic.
Right. What do these two specific chemical tweaks actually do in the human body?
Think of the original drug as a key that barely fits into a lock. It kind of wiggles around, but it doesn't turn well.
Okay, I can picture that.
Adding that highly electronegative fluorine atom, it pulls electrons in a way that radically increases how tightly the drug binds to its bacterial targets.
Oh, wow. So it just locks in perfectly.
Exactly. It supercharged the potency. And then the piperazine ring, on the other hand, changed the drug's physical properties. It drastically improved how well it absorbed in the gut and how deeply it penetrated into human tissues.
Tissues, like what?
Like the lungs, the bones, the prostate, places that are usually really hard to reach. Suddenly you had this pill that could achieve the kind of systemic, full-body bacterial eradication that previously required an intravenous drip in a hospital.
Man. So it turned a local tool into, like, a weapon of mass destruction for bacteria.
It really did.
But to really grasp why they are so powerful, we need to zoom all the way in on what these drugs are actually doing inside the bacterial cell. We need to look at the exact moment they interact with bacterial DNA, which involves solving what biologists call the packing problem.
Yeah, the packing problem is fascinating. So every bacterial cell faces an enormous logistical challenge. A bacterium's DNA is thousands of times longer than the cell itself.
Wait, really? Thousands of times?
Yes. So to fit inside, that genetic material has to be tightly coiled and then supercoiled.
Wow.
However, a cell cannot survive if its DNA is just permanently packed away like that. Whenever the bacteria needs to replicate or read its genes to build proteins, it has to temporarily uncoil that specific section of DNA.
It makes me think of an old, hopelessly tangled, coiled phone cord.
Yes, that is a perfect analogy.
Or like if you take a long piece of heavy rope and twist it against its natural lay, if you try to pull the strands apart in the middle, the twisting tension just builds up on either side until the rope knots up on itself and jams completely.
Exactly.
You have to find a way to relieve that physical strain, right?
And bacteria solve that exact mechanical tangling problem using specialized enzymes called topoisomerases.
Topoisomerases.
Right. These enzymes function as molecular stress relievers for the DNA. And fluoroquinolones target two of them in particular, DNA gyrase and topoisomerase V.
Okay, let's break those down, because the mechanism here is honestly kind of terrifying when you realize what's happening on a microscopic level.
It is pretty brutal. So starting with DNA gyrase, which manages tension ahead of the replication fork. Imagine a zipper being pulled apart to copy the DNA.
Okay.
The material ahead of the zipper gets overwound and incredibly tight, just like your rope analogy. DNA gyrase steps in, physically cuts both strands of the DNA, passes another section of the strand through the brain to relieve the tension, and then pastes the ends back together.
That's incredible.
Right. And without this enzyme, the replication process just stalls out because the DNA becomes physically impossible to separate.
And the second target, topoisomerase V, what does that one do?
Topoisomerase 6V handles the final stage of replication. So bacterial chromosomes are circular. When a bacterium finishes copying its DNA, it is left with two circular daughter chromosomes that are physically interlinked.
Oh, like two metal rings in a magic trick?
Exactly like that. Topoisomerase V performs another cut-and-paste maneuver, slicing one ring open, passing the other one through it, and sealing it back up so the cell can actually divide into two new cells.
Now here is where it gets lethal. Fluoroquinolones don't just, like, put a cap on these enzymes to stop them from working. If you're listening and wondering how a drug destroys a cell just by blocking an enzyme, it's actually way more aggressive than that.
It really is.
The drug waits for the exact millisecond when the enzyme has snipped the DNA strands open, and while the DNA is in that vulnerable cut state, the drug binds to the complex and traps it.
Right. Pharmacologists refer to this frozen state as a cleavage complex.
It's like waiting for someone to open a pair of molecular scissors and then jamming a rock into the hinge so they can never close them again.
That's exactly what's happening. The enzyme is completely paralyzed mid-operation. So what the cell intended to be a temporary, easily repairable break is suddenly converted into a permanent, gaping double-strand break in the genetic code.
And the bacteria can't just route around that. The sources explain that the bacteria's own replication machinery comes cruising along the DNA track at high speed, crashes directly into these frozen broken ends, and triggers a catastrophic fragmentation of the genome.
Yeah, it's a disaster for the cell.
The bacteria basically shreds its own DNA trying to fix the jam.
And this specific genome shredding mechanism is why fluoroquinolones are classified as bactericidal, meaning they actively and violently kill the bacteria.
As opposed to what?
Well, many older antibiotic classes are merely bacteriostatic. They just pause bacterial growth and rely on your body's immune system to clean up the sleeping invaders.
Oh, I see.
But fluoroquinolones take no chances. They initiate cellular self-destruction.
Which perfectly explains why they became the ultimate medical weapon of the late 20th century. Taking a highly bioavailable pill that actively shreds bacterial DNA anywhere in your body is an incredibly appealing concept for a doctor.
Oh, absolutely.
But reading through the prescribing data from the 1990s and early 2000s, that exact power led to massive unchecked overuse.
It did. The FDA's approval of ciprofloxacin in 1987 initiated what is now widely known as the Cipro era. It rapidly became one of the most prescribed antibiotics in both American hospitals and standard outpatient clinics.
Doctors were handing it out for simple bronchitis, traveler's diarrhea, minor sinus infections. I mean, conditions where older, narrower spectrum drugs would have worked perfectly fine.
Or where no drug was needed.
Exactly. Honestly, where no antibiotic was needed at all, since many of those are viral, it seems like it just became the ultimate drug of convenience.
Well, if we connect this to the bigger picture, why spend the time and resources figuring out exactly which specific bug is causing a minor infection when you can just drop a broad-spectrum nuclear bomb on it in pill form and send the patient home?
Right. And we saw the peak of this reliance during the 2001 anthrax letter attacks in the U.S. When those spores were sent through the mail, the CDC actually stockpiled and distributed millions of doses of Cipro to postal workers and Capitol Hill stash.
Yeah, and given the mechanism we just discussed, it was the logical choice for a bioterrorism threat. Cipro's ability to penetrate deep tissue and rapidly destroy bacterial DNA meant it was one of the only oral drugs capable of reliably preventing inhalational anthrax.
It undeniably proved its immense public health value in that moment.
It did. But the sheer volume of this drug pouring into the population came with a massive cost. We basically carpet-bombed society with this antibiotic for 2 decades.
And what was the fallout from that?
The primary cost was severe antimicrobial resistance. By using it for every minor sniffle, we exposed countless bacterial populations to the drug, allowing them to mutate and adapt.
Just basic evolution.
Right. We started seeing terrifying widespread resistance in pathogens like E. coli and Neisseria gonorrhea, rendering the drug useless against the very infections it used to cure easily.
So we have this massive paradox. The overuse eroded the drug's effectiveness, but simultaneously, giving this heavy-duty drug to tens of millions of people generated a gigantic real-world pool of mass exposure data.
And that data was revealing.
The math basically guarantees that if there's a dark side to a medication exposing that many people will bring it into the light, and the side effects that started popping up in the databases were completely unrelated to the antibacterial mechanism.
Right. Rare but devastating side effects suddenly became statistically undeniable, and they were occurring in human tissue, not bacteria.
The case reports on the tendons actually stopped me in my tracks. Patients were experiencing spontaneous Achilles tendon ruptures.
Just out of nowhere.
Yeah, not from sports injuries, but just walking down the stairs, sometimes within just days of starting the pill. And often bilaterally, both Achilles tendons snapping at the exact same time. I mean, how does a drug designed to kill bacteria cause a human tendon to physically snap?
The mechanism is deeply unsettling, and it points to severe off-target effects. Fluoroquinolones appear to interfere with human collagen synthesis and upregulate the activity of matrix metalloproteinases, or MMPs.
Hold on, what? Are those just the enzymes that normally maintain our tendons?
Essentially, yes. MMPs are enzymes that break down proteins. They act as the body's natural recycling crew for tissues, clearing out old material so new tissue can be built.
Okay.
Fluoroquinolones throw this recycling crew into absolute overdrive. The drug causes these enzymes to rapidly degrade the structural collagen fibers of the tendon itself, weakening the tissue from the inside out until it reaches the point of catastrophic structural failure.
Wait, if the drug degrades collagen matrices, is that why we also saw the FDA issue warnings about aortic aneurysms? Because the aorta is heavily dependent on collagen to handle the pressure of blood leaving the heart. Right.
That is the exact physiological link, yes.
Yeah.
The aorta handles the highest blood pressure in the human body. If the collagen lining that artery is rapidly degraded by those same upregulated enzymes, it creates a weak spot.
It's terrifying.
It is like a garden hose with a thin spot expanding under pressure until it tears an aortic dissection. That is a highly lethal event.
And the regulatory history here is just an escalating series of red alerts from the FDA trying to catch up to this damage.
The warnings snowballed over a decade. In 2008, the FDA added their strongest possible warning, a boxed warning, specifically for the risk of tendinitis and tendon rupture.
But then I look at 2013, and they had to mandate label updates for permanent peripheral neuropathy. We're talking about severe nerve damage here.
Yeah, life-altering nerve damage.
Patients reporting burning, tingling, and weakness in their limbs that didn't go away even months or years after they stopped taking the medication.
The true regulatory paradigm shift, though, occurred in 2016. The FDA issued a sweeping communication explicitly instructing doctors to stop using fluoroquinolones as first-line treatments for acute sinusitis, bronchitis, and uncomplicated UTIs.
So they finally said enough is enough.
Exactly. They officially declare that for common, self-limiting infections, the risks of this medication vastly outweigh the benefit.
They didn't ban the drugs, but they effectively said, only use this if the patient has absolutely no other options, and the hits kept coming. By 2018, the FDA added boxed warnings for dangerous drops in blood sugar. The brain relies entirely on glucose, so these sudden drops were literally triggering comas in diabetic patients. They warned about severe mental health disturbances, memory impairment, delirium, disorientation.
It's a huge list.
They even warned it could drastically worsen myasthenia gravis, leading to respiratory failure. Plus, QT prolongation, where it messes with the electrical recovery cycle of the heart and triggers dangerous arrhythmias.
It represents one of the most extensive and severe warning histories of any modern outpatient drug class.
And we absolutely cannot talk about this regulatory reckoning without acknowledging the patients who drove it. The sources show that patient advocacy played a monumental role here. A grassroots movement formed of people who developed these persistent multi-system symptoms. They started calling themselves Floxed patients.
These patients actively lobbied the FDA for years. They compiled data, they testified at hearings, and they coined the term fluoroquinolone-associated disability, or FQA. It is a striking example of how public pressure and relentless patient reporting can force regulatory bodies to reshape drug safety policy, often long before scientists in a lab have ironed out every single mechanistic detail of why the damage is happening.
But it raises an incredibly important biological question. If this drug was engineered so specifically to target a bacterial topoisomerase enzyme, how on earth does it end up shredding human nerves, destroying tendons, and causing cognitive delirium?
That's a great question.
Because here is where it gets really interesting. The answer forces us to look millions of years into the past at our own evolutionary biology.
It hinges on the endosymbiotic theory. So every human cell contains mitochondria, which operate as the energy-producing powerhouses of the cell. But mitochondria were not originally human.
Right.
Hundreds of millions of years ago, they were independent, free-living bacteria that were engulfed by our ancient ancestral cells in a mutually beneficial merger.
So our cellular energy factories are basically ancient domesticated bacteria living inside us.
Exactly. And because of that ancient bacterial ancestry, human mitochondria still retain their own distinct circular DNA, entirely separate from the DNA in our cell nucleus.
Oh.
More critically for our topic, mitochondria possess their own topoisomerases to manage that DNA, and those enzymes are structurally very similar to the bacterial gyrase that fluoroquinolones are built to attack.
Mind blown. So the leading hypothesis is that all these severe toxicities, the nerve damage, the tendon degradation, the cellular fallout stem from the drug accidentally hitting our mitochondrial DNA.
It is a massive case of biological friendly fire. The drug is perfectly executing its bacterial killer programming, but it is mistaking our own mitochondrial machinery for a modern bacterial invader.
That makes so much sense.
The very evolutionary history that allows the antibiotic to work in the first place is the exact same mechanism that makes human cells vulnerable to its toxic side effects.
The friendly fire makes total sense when you realize the friendly targets look almost exactly like the enemy. It completely reframes how you look at a pill.
It really does.
But before we wrap up, there are a few misconceptions floating around that we need to clear up. Hearing a list of side effects that includes ruptured aortas and comas, a listener might assume these drugs were just yanked off the market entirely.
No, they remain FDA approved and widely available in pharmacies worldwide. However, their role has shifted from a careless first resort to a strictly reserved second-line option.
Right.
We still desperately need them for severe, life-threatening, or highly resistant infections, like complicated intra-abdominal sepsis or bioterrorism threats, where safer alternatives simply will not cure the patient.
Another thing people might assume is that these catastrophic side effects only happen to the elderly or people who are already severely ill.
That's a common misconception.
Hmm.
While being over 60, having a history of organ transplants, or taking concurrent corticosteroids does significantly elevate your risk, the pharmacovigilance data is clear. This is not just an aging issue. Yes, completely healthy young adults and even elite athletes have suffered from spontaneous tendon ruptures and disabling neuropathy after short courses of these drugs. It is highly unpredictable.
And I want to quickly push back on a fallacy that always pops up when we talk about lab-made drugs. People hear fully synthetic and immediately assume that's why it's toxic. The whole natural is safer argument.
Ah, yes.
But plenty of natural things like botulinum toxin will kill you instantly, and plenty of synthetic things are perfectly safe. The toxicity of fluoroquinolones has absolutely nothing to do with being engineered in a lab.
Correct.
It is entirely due to specific biochemical off-target hits on collagen mitochondria. The chemistry is doing exactly what it was built to do. It's just hitting unintended ancestral targets.
Synthesizing all of this, fluoroquinolones stand as a critical pillar in understanding modern medicine. They sit right at the collision point of two major global health crises:
Which ones?
the terrifying rise of antimicrobial resistance fueled by overprescription and the complex challenge of long-tail, hard-to-predict drug toxicity.
They really are a living, breathing cautionary tale. Every time someone takes a Cipro pill today for a minor urinary tract infection, they are participating in a regulatory framework that was paid for by 20 years of accumulated harm reports from patients who are told the drug was perfectly safe.
It is a stark reminder that drug safety science does not end on the day a medication receives FDA approval. It evolves in real time, in the real world, and often with profound human consequences.
Which leaves you with a final thought to mull over, building on that mind-bending evolutionary connection. If our mitochondria are so closely related to bacteria that standard antibiotics can accidentally damage them, how many other things are we missing?
That's a profound question.
How many seemingly unexplained issues in modern society, like chronic fatigue, metabolic disorders, or mysterious joint problems, might secretly be tied to the low-grade, off-target mitochondrial impacts of the everyday medications we take completely for granted? We started out talking about precision weapons and load-bearing walls. It turns out, when the weapon targets an enemy that is literally woven into the evolutionary foundation of your own cells, there is no such thing as a clean strike. Thank you for joining us on this deep dive. Keep asking those hard questions, keep looking past the label, and never stop digging deeper into the science around you.